Welcome, everyone. I'm Penny Manasco. I am the CEO of MANA RBM, and we are happy to have you here. We have a full house, and we are excited to be able to share this training program with monitors and clinical research professionals around the country. Let's get started. This is one in a series that will go throughout January and February of 2016, and hopefully we'll give you some tools to be able to start adopting risk-based monitoring within your organization. So why MANA RBM to be the ones to teach you? First of all, I speak a lot around the country, and one of the things that I hear is tell us how to do this. So it's great to have theory, but we really want practical advice on how to do this. So we feel at MANA that we are the perfect people to be teaching you because we have been working on the principles of risk-based monitoring and remote trial management our team has for more than 10 years. So I'm a physician. I became a physician specifically to do clinical research. So this is my passion. And I believe risk-based monitoring is one of the most important things to happen to the area of clinical research in my entire career. I think it's was great insight from the regulators, and I think that this is going to make a huge difference in the way we're able to conduct trials. Just so that you know, I have done pretty much every job. I've been a PI at the NIH. I've been a vice president at GlaxoSmithKline. I've been a chief medical officer at 2 different technology companies, and those were small companies. So I did a lot of different jobs within those companies, including designing CRFs and all kinds of different activities. I have been trained and I have been boarded in both peds and pediatric endocrinology. My team has worked with me to develop and implement the entire process of risk-based monitoring, and we've done this for multiple sponsors. We have also completed a knowledge transfer to a sponsor. So we believe that we don't have the only answer, but we have an answer. And that really allows us to help you to give you the advice that you need on how to implement RBM, whether it's little pieces or whether it's the entire process. We've also led the process to develop competencies for monitors, and we've published that. And we've also published a number of papers on electronic investigator site files, remote informed consent review. We've done training at the DIA, so many different activities, all with the goal of trying to advance the area of risk-based monitoring. So these webinars that we have designed for you are designed to help monitors to adopt the principles of risk-based monitoring and begin self-training. So one of the things in our careers is we're trained one time and then we have to keep training. And so this is hopefully we'll give you the tools that you need so that you can begin to train and teach with this core of knowledge, be able to teach yourself or go out and start to find other sources for more individualized training. We want to develop a cadre of trained monitors that can implement RBM whenever we go out and talk about it. One of the questions is always, do monitors know how to do this? And we believe that monitors can be trained. And so we encourage you to continue the training, and if you know others, encourage them as well. We also want to enhance clinical trial quality, and we want to drive adoptions. So the videos for our webinars are going to be available through our YouTube channel, manarbm.com. And you can also subscribe and follow us on MANA Risk-Based Monitoring on Google+. And we will also, starting in February, have additional documents available on our website, MANA RBM. It's going through a revision right now, and as soon as that's available, we'll let you know. So the goal for the entire program is to help you to understand risk-based monitoring principles, the goals of risk-based monitoring, and how to achieve those goals. For today's session, what I'd like you to be able to do is to understand the background for adoption of risk-based monitoring and to understand the critical regulatory guidelines and how they're going to affect monitoring activities. So we'll each, as we'll see, um, risk-based monitoring, if it's done what we think is optimally, then you're going to have changes in three main areas. You're going to have changes in your processes, and we'll show you some process changes that you may want to adopt right away. We'll show you there will be changes in technology that will optimize implementation of risk-based monitoring, and that there are new skills and maybe even new roles that will be expected of clinical research professionals. So just to give you an idea of what the rest of the sessions are like, session 2, which will, each session is on Friday at 1 p.m. Eastern Time, and session 2 will help you to understand risk assessment, root cause analysis, risk mitigation, and corrective action. To me, this is one of the most important areas for you to adopt, and you can do this regardless of what's going on within your organization, because what this session will be is around thinking about the risks, identifying the risks, and understanding how you can correct those. Session 3 will be on understanding the different types of e-clinical technology available and how that data affects the monitor's roles. Traditionally, technology has been something we've kind of given off to the data managers, and what I want you to do is realize there's a lot of really important information, and you do need to understand some things about technology for your individual trials, as well as maybe if you are a more senior person, how all those pieces of technology will work together. Session 4, the monitor will be able to discuss lessons learned in implementing RBM. We're going to try to bring some of the things that we've learned and help you to see how different things that we've found in our methods will help you to adopt. We're going in session 5, we'll understand the differences in central monitoring and site monitoring and how the two interact. You'll hear that there have been publications about the potential role of a new central monitor, and we want you to understand how we at MANA have adopted that. Session 6 will be to understand the key components of source data review, including interpretation and analysis of critical data, and we'll show you some of the tools we've developed there. And finally, session 7 will be able to interpret different site performance. The monitor will be able to interpret different site performance data. So we'll show you some different ways to look at data and how that might indicate different ways that you may want to monitor and oversee the site performance. So these are the sessions. Will there be sessions after that? We don't know, and we'll be looking to you to find out if there are different additional things that you would like to learn as part of your knowledge quest. So what we would like to do as you go through this process is to think about how you're going to learn. So take your questions, spend some time thinking about different options, and hopefully that you'll come through this program with a number of aha moments. So we would like you to be able to see these principles that we're talking about. Many of them have been around for a while, but not in clinical research. Many of the things that we're going to talk about are a change from the way you learned clinical research. And so this process of kind of integrating it and understanding it may not come overnight. And this is why we want you to be able to have time to think about it, ask questions, and realize that we're here to be a resource for you. So risk-based monitoring really starts with quality by design. So quality by design started in the automotive industry, and the idea was think about what can go wrong and plan for that and figure out how you're going to eliminate that risk or how you're going to identify that risk and correct it if it does happen. So as we said, it started in the automotive industry. It's been used in both pharma and device manufacturing, and now it's coming into clinical development. So I like this picture because it gives you a way to think about just with one little activity, what could go wrong. So if we look at these gentlemen who are standing some area above the ground, standing on a ladder, hopefully holding the gentleman that is over the ledge and fixing the air conditioner. There are a number of different things that could go wrong. And what we're talking about in risk-based monitoring is for you to be thinking about whether it's data quality, whether it's subject safety, whether it's protection of the investigational product. All of these areas you need to think about what could go wrong and how are you going to know if it does go wrong. Now, this looks like a boring slide, but it's an important slide. It's important for a number of reasons. So first of all, these are the guidance documents from the FDA on the oversight of clinical investigations, a risk-based approach to monitoring. The EMA, European Medicines Agency, the reflection paper on risk-based quality management in clinical trials. And just recently, the ICH came out with their integrated addendum to ICH E6R1, which is the guideline to good clinical practice. And in this document, which is right now out for comment, but will likely, as I've heard, and again, I don't have this from the ICH, be adopted in 2016. So the bottom line, though, is all of the regulators are saying the approach that we've been taking to look at trial quality has been inadequate, and we need to improve the methods that we use. So this is not something that is going away. And so as I often tell people, when you are trying to advance your careers, one of the most important things you can do is to learn new areas, especially if they're not a fly-by-night area, but something that's going to be important. So I congratulate you all for being here. Now, these documents that are in black font, these documents are available for you to download during the webinar. So you can pull these documents right from the webinar and have them for your reference. There's also the Q10, which is the FDA's Pharmaceutical Quality System document from 2009. And although this has a lot of manufacturing, it is a good reference document for you. About the same time as the risk-based monitoring guidance came out, there was also guidance that came out around the use of electronic source. An electronic source is the idea that the first place that you collect that data is considered the source. Now, as much as possible, if we can make that electronic and collect that the first time we see a subject, then that's optimal. And we'll go through a little bit about that guidance in today's session, but we'll go through it much more in future sessions. And this idea of electronic source comes from both the FDA and the EMA. And there's another document that I think is really important, and that's 21 CFR Part 11. And we'll talk a little more about that today. This has to do with regulations for electronic systems around e-signatures. So and it is an important document. And then there are additional documents around use of electronic informed consent for clinical investigations that was released this year as well. So these are just some regulatory guidance documents. Again, you may know these. If you haven't, read them because I think that they become an important basis. And just as when we design the way that we're going to look at our safety oversight, we look at the guidance from that the FDA reviewers get as far as how they're going to do their safety review. The same kind of idea, if you understand these documents, then you're going to be much better able to implement them. So what does risk-based monitoring mean to the course participants? First of all, it can mean, to some people, it means reduced SDV. To others, it means reduced on-site monitoring. Those are the most common ways people have heard about them being adopted. Some people talk about risk-based monitoring as just reduced number of data points and or patients being monitored. In other words, 100% of SDV for 10% of the subjects plus 100% of SDV for AEs or inclusion and exclusion criteria. Some people think of it as reduced on-site monitoring offset by an increase in remote monitoring, but really what risk-based monitoring is the reduced on-site monitoring offset by an increase in remote data review and analysis. It's really important because we're getting away from just looking at data points and checking data points to really thinking holistically about what the data are telling us at an individual subject level, at a site level, and across sites. So let's start with the guidance and get into it in a little more detail. The first point is the overarching goal in the FDA guidance was to enhance human subject protection and the quality of clinical trial data by focusing sponsor oversight on the most important aspects of study conduct and reporting. Okay, so it's about trying to focus resources on the most important areas. And why are we doing this? Why did all the regulators come out with this guidance at the same time? Because despite 100% SDV, the most common FDA finding during sponsor monitor inspections has been inadequate monitoring. And these are the results from 2014, but they could be the result from pretty much any year. So number one is inadequate monitoring, failure to bring investigators into compliance, inadequate accountability for investigational product. You can see IP is right up there in the very important top areas, and failure to obtain FDA or IRB approval prior to study initiation. So these are what we're going to be focused. So this is why we need to be focusing on how to do a better job of monitoring. So the consequences could be disastrous and have been disastrous. So it could be as simple as as having to send an army of people out to a few sites because the data are a mess, or you didn't have a good monitor, or you think that this is a site that's going to be audited as part of your registration. So you could have all of that resource that you have to spend there. I've had sponsors telling me that they've had to lose an entire site because the quality of the data on the primary efficacy endpoints was not of sufficient quality to be able to use the data. And this was 56 subjects that were treated and their data not used, which is, I think, putting subjects at even greater risk. But the stories go on. Everyone who's been doing clinical research for a few years knows about the consequences of missing important aspects of monitoring. But they also could be 483s. You could have your drug or device not approvable. You could have your drug or device withdrawn from the market because safety issues were not really discovered or recognized. Deaths become because risks present all along were not discovered until the product went to market. So sometimes there could be signals there that had been missed, and sometimes the study has to be repeated. So these are all pretty significant consequences. Now, let me give you some real life examples to help you to put this into perspective. So these came out in the 2009-2010 time point. So one was a 483 that Pfizer got for failing to ensure proper monitoring for a trial. This was a pediatric trial, and the subjects were consistently misdosed. And this went on across sites. It went on despite monitors being at the site on more than one occasion and doing 100% SDV. Now, are these bad monitors? No, we have set monitors up to look at data as part of SDV in a very much a point-to-point or in little uh, pieces, kind of sections of information, and not giving them the tools that they need to be able to look more comprehensively. So in this situation, and I was at a risk-based monitoring seminar about a year ago. And in that seminar, Jonathan Helfgott from the FDA also spoke, and he mentioned this case specifically and talked about the fact that it took way too long to find out that the subjects were being misdosed. I believe it was identified finally by a data manager, but months after the subjects had been started to be enrolled. In addition, I think he mentioned that there were some tools that weren't available to help the sites to better uh, plan their dosing. So a number of things went wrong, but the bottom line is it did affect both investigational product and subject safety. The second example was J&J and Icon, and they were cited for not identifying suspected fraud in a study drug administration. And this was also mentioned under the data monitoring deficiencies, ciftobiprole, and this was in the U.S. once again. So these are the references for these papers, and data integrity, subject safety, investigational product. These are what the FDA and we all recognize as the really important areas that we need to spend time on. So a concept that you'll hear is not all data are created equal. Some data are more important and need to have more focus than others. So these are real life examples, and hopefully the approaches that we're going to teach you over the next 2 months will help you to be able to avoid some of these. So some of the other pieces that are important here are that multiple approaches are acceptable. So as I mentioned earlier in the presentation, there are many ways that you can do risk-based monitoring. What we'll talk about is our methodology, just because it's, as we said, many people want to get real life ideas, real life details, and not more theoretical. And so it's very difficult if you don't have experience across all of these broad areas, whether it's safety, monitoring, data management, pharmacovigilance, all of these. If you have a team working together, that works, but it's very difficult to really understand how all these pieces fit together. So we're going to talk about what we call the MANA method, but it's only one method to do risk-based monitoring, but hopefully it will give you ideas. The FDA also recommended greater use of centralized monitoring methods, so remote data review and analysis. They also wanted to focus on critical data elements and critical processes, and we'll talk about that more, and that electronic systems enable remote, near real-time review of data and documents. So this is an important piece because we want to be sure that we start looking at data as quickly as possible. Oh, let me go back here. So when is RBM not appropriate and why? So do you use RBM in phase 1 studies? Do you use it in phase 2? Do you only use it in phase 3? Do you use it in complex studies but not oncology studies? The answer is you should use risk-based monitoring in all of your trials. So all trials are appropriate. The number and frequency of on-site visits may be different. What you do at the on-site visits may be different. But what we're talking about is oversight. So oversight, these principles of focusing on the important data for data integrity, for protocol compliance, for GCP, for subject safety, for investigational product, you're going to hear me talk about those over and over again. These are the areas that we really want to focus our oversight on. So this is a direct quote from the guidance, and let me just, I'm going to go slowly over this because it is very, very important. So in that guidance, the FDA says that the risk-based monitoring approach can enable a reduction in SDV, but it doesn't suggest any less vigilance. And one of the things that we hear and over is, if we don't do SDV, we're not doing a good job of quality oversight. And we're going to show you how you do quality oversight without SDV. Rather, it focuses sponsor oversight on preventing and mitigating important risks to human subject protection, data quality, and trial integrity. Moreover, a risk-based approach is dynamic, more readily facilitating continual improvement in trial conduct and oversight. So that's an important piece as well, as we need to learn from errors, we need to make sure to correct them. So that's all part of the issue management and root cause analysis that I mentioned earlier. The FDA encourages greater use of centralized monitoring practices where appropriate, than has been the case historically, with corresponding less emphasis on on-site monitoring. So what we'll be doing is showing you what you do when you are not doing your monitoring at the site. So guidance Schmidance, do we have to follow it? And the reason I put this in is that I talk to sponsors all the time. Risk-based monitoring document, this risk-based monitoring guidance document is only a guidance document. So do we have to follow it? Well, the answer is really no, you don't have to. But if you think about all of the different guidance documents that we use to guide the way we do our development plans, the way we do our safety reporting, you realize that regulators very seldom, and I'm going to give you an example where they did require, but very seldom do they give you firm requirements. And what they do is give you their best thinking at the time, and you realize that it's often in your best interest to adopt the kinds of guidance that they provide. So, as I mentioned, one of the areas that was also included in the guidance documents and is also a guidance document is the idea of electronic source. And the reason this is important is that if as much as possible we can use the data directly as it's collected, and as much as possible have that in electronic format, then we are much better able to start our review quickly, identify problems quickly, correct them quickly. So I liked the principles of Alcoa. You'll find these also in the Society of Clinical Data Management's white paper on risk-based monitoring as well. Is what the FDA wants is they want the data attributable. And so some of the electronic source systems are nice because you know if the data are collected at the time that a subject is seen in the clinic, then you know who actually saw that subject as opposed to who transcribed the data. So it's really nice to know who actually saw the subject, have the data be legible, have it be contemporaneous. So that's the C, contemporaneous. And that's really important because that allows us to do a better job of safety oversight. Have it be original, again, not transcribed, and have it be accurate. So these are the principles of eSource, and we'll talk a lot more about eSource in our technology section, but I do want you to know that this is a term that you'll hear, and it can mean it is any electronic document where the data are collected the first time. So that could be a blood pressure measurement, it could be from a lab, it could be a medical record for things like medical history and concomitant medications. There are a number of different, and it could be your EDC system if you have the sites collect the data contemporaneously, or it could be a separate eSource system. So there's a lot of different ways that you can reach these goals and a lot of different price points too, which is important for you. So 21 CFR Part 11 is a different document, but it is not a guideline. This is a regulation. That's what the Code of federal regulation means, okay? And this is an important one, even if you are a monitor or a clinical person, and let me explain to you why. Um, First of all, this document has important regulations for e-signatures. And one of the things that we have in our training on 21 CFR Part 11 is it includes two pieces. So there's a piece from the technology standpoint. Does a system have certain capabilities? Okay. And then there's a process system. So whenever you have someone say, oh, my system is 21 CFR Part 11 compliant, you need to question that. Because you have to have process as well as technology to meet the guidelines for 21 CFR Part 11. So some of the pieces that you need to know about are, again, for e-signatures. Um, First of all, you need to know that in 21 CFR Part 11, they require that any company that uses e-signature send a letter to the FDA. So if you don't have to do it for every trial, but you do have to do it at least once for your company. Your e-signatures must include attestation language. That means I am certifying, if it's for, let's just say, you probably often seen it, that a PI signs, I certify that I've reviewed the data on these ECRFs and they are correct. But it could also be used as we do when we're making certified electronic copies, and you can say, I attest that I've reviewed these two documents and they are the same. But that language needs to be in there. You need to include two methods of identification. It must be attached to the document permanently. So when you PDF that document, the e-signature with the audit trail, really, which includes the date and time and user information, is all included in there. So e-signatures become a very wonderful, usable tool in lots of different ways, not just for the PI to sign at the end of the study. And so it's important to know this. But there's also regulations on validation, on audit trails, and on training. And the reason that I bring this up is, first of all, from a monitoring standpoint, the audit trail can be a really useful tool for you. And you need to recognize that training and documentation of training and keeping track of that is an important aspect of 21 CFR Part 11. Everyone who uses an electronic system must be certified that they are trained, and you must have that certification. So it's not usually a problem at the beginning, but it is often a challenge as your study goes on, especially if it goes on for a long time. So you want to be sure that you're keeping track of that and realize that this isn't a guideline. This is a real regulation that you need to follow. And we'll talk about more on that in our session on technology. This is just to give you the reference for the competencies. We worked with a wonderful group that included people who had come from the device industry, pharma and biotech, who had come from academic research organizations, who had come from clinical research organizations, and who were based in India, Europe, and the US. So what we wanted to do was to get a very broad view of how the monitor's role will change as risk-based monitoring is implemented. And we also developed a competency survey. So here's the URL for that, and you can go on and you can, we've developed a bunch of questions. These are better done on a laptop than on your phone. But you can take this and periodically we review all the results from people who have submitted them, and we will give you your results back so that you'll know what areas you did well in and what areas you might want to work on. So let's just go through the competencies, because this is where we want you to get to by the end of this. One is that monitors will be able to evaluate clinical data from individual subjects to assess whether that subject is appropriate. So moving beyond SDV to source data review. To evaluate and analyze clinical data across subjects and across sites. So we'll talk more about that, because that's a process that hasn't been incorporated routinely in the clinical research process. to be able to perform centralized review and interpret and analyze site performance in comparison with other sites, to proficiently use clinical trial technology. That's why we've included that as a specific session on the webinars, and to develop consistent strategies for assessing and resolving problems, including root cause identification, remediation, and evaluation. And that's what we'll be talking about next week. Traditional monitoring. So traditional monitoring today has been focused on source document or source data verification. The D could be either way, with little critical or analytical review. So SDV versus SDR, and we'll talk more about that in a minute. But this is probably the hardest thing for people who have been in the industry, is that what we're saying is the way that we've been doing trial quality oversight has not really been integrated enough, and so we need to step back and figure out what we do. And so the critical elements to ensure integrity of a clinical trial, and that is, do we have complete data or all the assessments there? Is the subject safety being appropriately reviewed? Are dosing and IP management being reviewed? Or do we have full and complete review of informed consent? Informed consent is often done, but many of these other areas are often overlooked. And even though informed consent is, quote unquote, already done, it is still one of the top 5 findings from a site standpoint when they have their audits. So this is what we will be focusing on these critical data elements and processes. So Transcelerate is a group of pharmaceutical companies. Not sure how many there are now. They started, I think, at 11, and they've continued to grow. They do a lot of internal sharing of pre-competitive information, but they've done a very good job of providing some very good basis and very good background in the area of remote risk-based monitoring. So I would recommend you go to their website and review the documents that they have. We'll be talking about some of them during the webinars, but it is a good place. And one of the things that they did early on was to differentiate between SDV, or source data verification, and SDR, and source data review. So what they did is defined SDV as really being transcription checking, and that is what the vast majority of monitor time is spent on. And if you ask monitors, which we do all the time, what about reviewing documents? What about reviewing IP? What about time with the PI? Many times they will tell us that they have not been able to do that because they were too busy doing SDV. And so what we want people to begin to think about is, what is that best use of your time? Not that we need to spend more time, but we need to spend the time differently. So source data review puts the focus on review of source documentation in the context of a comprehensive review of the quality of the data. So it is not a comparison of, it's not transcription checking, but it's a more synthesized approach. And as I said, we've developed some tools to help there, because we think this is a really important change in the way that you look at data, and this is something that we can teach monitors to do very efficiently, and we'll get a much better outcome than the current approach. So let's talk a little bit about SDV and SDR, because I think that for some people, I mean, we've been talking about it for such a long time, we understand it, but what I want to try to do is give you some other ways to think about it. So if you were to be doing SDV of these two data sets, if you will, what you would be doing would be checking to see that you had exactly the same dots in the same place, that they were maybe the same color, so basically a transcription checking of one compared to the other, okay? Now, what we're saying is we want you to step back and do a more comprehensive review of the entire picture of the subject. So if you think back, go back here a minute, you're maybe in visit 1, you're just checking this amount of data, and then in visit 2, you're checking just this amount of data, and in visit 3, you're checking just this amount of data. But if you're not thinking about looking across all of these, then even when you do SDV, you can see you're not really able to compare and see things that may look funny from one visit to the other. So all of this, when you are just doing point-to-point checking, you miss that ability to step back and look at the total picture. So why don't we do SDV? So it's tedious, and I know many monitors really hate doing it because it's pretty boring. But it's easy to teach people how to do. So the nice part is, as we've needed more and more monitors to go out to do the site visits, as this has been the expected norm within the industry, you can train a lot of people to do this. And it's much harder to train people to think about looking at the data in a more comprehensive, synthesized way. So that's why I think that a lot of times it's easy to train people to do it, but it's very expensive. And monitoring costs are the second highest cost of after investigator site payments in trials. And so they really drive the costs up at the trial. And much of the time that the monitor spends, so if you look at the traditional numbers, and these I've checked with many people, is that for an on-site monitoring visit, there's 21 hours of time usually assigned. So of that 21 hours, that's for an 8-hour on-site monitoring visit. So yes, some time is spent on reviewing the data in the EDC, sometimes, but in general, much of that time is really spent in travel and not in doing monitoring. So what we want to do is to really change that dynamic and have that time that was spent in travel and all these other things really spent on looking at the data and reviewing the data from the subjects. And I'm going to show you in a minute why we think this is inadequate study oversight. So this is a paper from the group at Transcelerate. This was published in November of 2014 in, let's see, Therapeutic Innovation and Regulatory Science. And it's a fascinating paper because they looked at 1,168 trials across a number of sponsors. And what they found, and this was, I believe, done through Medidata, but they looked and found that only 3.7% of the data ever changes. So 96% of the data that's entered by the site never gets corrected by any method, okay? And of that data that is corrected, only about a third of that could be attributed to SDV. Now, of that, we don't know what percentage of that was a critical data element for safety, for efficacy, for investigational product. How much of it could have been found another way? So the bottom line is that if you looked at the millions of dollars spent for the amount of value you get on just SDV corrections, you would see that this is not a good use of resources, and that's why we've moved to the more risk-based monitoring approach. So here are some misconceptions about RBM. One is that you might miss something if you don't go out to the site and you don't do 100% SDV. And there's a couple of points there. Let's go out to the site. So first of all, we think you should go out to the site. We think you should go out to the site when you do the initiation visit, because what you want to be doing is building a relationship with that site so that in the future, you can build on that relationship with remote methods. So you have to start by building the relationship with the site up front. And you want to be sure that they're trained on the methodology and that they understand what you're going to be doing and how you're going to be doing the monitoring. Because oftentimes we hear sites being very concerned that they're going to be out there on their own. Nobody's going to be helping them. Nobody's going to be reviewing it. And that couldn't be further from the truth. So first of all, you do go out for site visits. You just don't go out as often. And again, what you do at the site visits can be very different. So um, even if you even if you don't do 100% SDV, you should be finding more issues because of the way you're going to be looking at the data than when you were just doing SDV. So studies have shown that there's little value in SDV and that careful review and analysis of the data done remotely is much more likely to uncover protocol inadherence, lack of understanding to the protocol and or assessments, toxicities, and other potential problems. And you'll see this in the Sheets article here, which we have on the slides. Some other misperceptions about RBM. One is that the only change in RBM is that there's less SDV. And we talked about that at the beginning. That's not the case at all. What we want to do here is we're changing from SDV to comprehensive source data review and review at different levels that have not even been considered as part of risk-based monitoring. So the second one is with implementation of risk-based monitoring, study monitors will lose their job. And that is not true. Monitoring will be different. It's going to be more analytic. It's focused on critical data and processes. You'll be spending, the time on understanding why errors are being made by determining root cause and corrective action. And site interactions are still critical. So it's really, really important to realize the site monitor can't be more important, but I believe the site monitor is going to be a much more significant contributor to the oversight of quality than they have been from a content standpoint. Data managers will lose their job. That's another misperception. In fact, one of the things that we have found really powerful with our approach is the much closer working with data management and the clinical monitoring team. So each group looks at data a little bit differently. They think about it. They think about the impacts differently. And so what we think is that what you'll see is more of a removal of some of the silos and a more close working relationship. The last one that we hear is that risk-based monitoring will put smaller CROs out of business. And I don't believe that at all. I believe that smaller CROs, small and mid-sized, really have a great opportunity here to effectively adopt risk-based monitoring. We think they can be more competitive. We think that they may be more flexible in the way they're able to adopt these kinds of approaches and better use their staff that they have and maybe have less worries about the quality as they're able to do more oversight remotely. Now, over the time, we're going to talk a lot about some of the process differences. So first and foremost is the risk assessment and quantification. So I think this is the most important thing you can do of pretty much everything, because if you are thoughtful and think about first, how can you remove the risk during protocol development? And then once you've finished your protocol, what are the risks, remember, in the critical areas? What are the risks in protocol compliance? What are the risks in GCP? What are the risks in subject safety? What are the risks around investigational product management? And data integrity. So I think that if you can focus on your efforts on those and understand what the risks are, you can begin to plan. And that's what we'll talk about. Is a lot of this is about thinking about, now that I understand the risks, how am I going to identify those? Who's going to look at those? And that process, and we'll talk about that next week. Review timing, I'm going to show you a slide about that. I think this is something that if you want to take home, when I was in med school, in our lectures, we used to call them pearls. If you want to take home a pearl, this is the next one is a really good pearl that you can take home and that can make an immediate difference in the way you do your trials, and that's review timing. And documentation oversight is another important area. Much of this can be done remotely now. We can do a much more, we can be sure that investigators have all the documents that they need, which helps to improve audit performance. Investigational product oversight, how we can do investigational product oversight remotely is another area that I think can really help you, so that you, doesn't mean you don't do it when you go out to the site, but that you can keep a better handle on it while you're working remotely. Again, when do you use SDV? How do you do it? And how do you document that in your monitoring plans? We use what we call an integrated 3-tier review, and I'll show you a picture of that in a minute. But the idea there is that there is review done by the monitor and data manager, again, right away, and then a higher level review that comes after that. And then finally, the type of data that's reviewed. So we talk about high-risk data elements. What are those specific areas that are going to be important from an efficacy standpoint, from a safety standpoint? How are we going to look at those? Operational metrics. So we look at, for instance, queries, but we look at them differently. So we're going to be looking at how many queries do we have? Who are the people that are having the queries? Are we seeing questions that either indicate lack of understanding? All of these kinds of areas we want to look at. Not just are the queries closed so that we can lock that subject. Same way with audit trail data. Oftentimes with eSource, we want to look at that because it gives us an indication of who is having a problem, not just that the site is having a problem, and that allows us to be much more granular in our and more focused on our interventions. So one of the other learnings that we've had, and I think will be important for you to understand as you have these communications within organizations, is that when we talk to groups of BDS, so biometrics and statistics, they say, we have all of the data from the trial. We get that from the CRO. But what we often find is that data has been converted to SDTM, which is a format. So if you're from a clinical standpoint, you may or may not know this. This is CDISC, the Clinical Data Interchange has developed a format with the FDA that they want data submitted. And when they do that, much of the data, such as the audit trail and the query trail and all of these things, are kind of merged and buried and not as useful when we're doing risk-based monitoring. And then one of the other things that we think is important is an integrated view of data across data sets. So some of these we'll talk about today, but we'll cover these in more detail in future seminars. So this is this came out of a metadata blog, and I want to walk you through it carefully. So the red is the time from median time from subject visit to query close. Now this includes automated queries as well as manual queries. And you can see in 2009, that time was 36 days. That was the median time from visit to query close. They got it down in 2011 to 30 days, or slightly less than 30 days. But then it was back up by 2012 into the 30 and 35 days. So to me, that's important, but it's difficult because of the automated queries to know what this really means. But the more important one to me is the one in green, and that is eCRF entry data to data management query opened. So this means that the first time the data manager probably looked at the data to be able to raise queries, and we're going to use this axis here, you can see that it was 90 days. So 3 months after that subject was seen was the first time that we started to get data management review. Okay, and that's because most processes require that the monitor go out to the site and quote unquote SDV, well, not quote unquote, and SDV the data before the data manager can review it. Now, so this is a tremendous opportunity right here to begin to look at data much, much sooner. And you can see they were able to get it down to 60 days in 2010, around 60. So that's still 2 months. But even by 2012, it was back up closer to 80 days. So what we're saying here is this is entirely longer than it needs to be. And this allows you to start to make an impact by looking at the data by the data managers much sooner. So let me show you this in another way, just so that I can drive it home. So again, we talk about the MANA method. That's just our approach. And again, it's approach to risk-based monitoring and remote trial management that allows you to see ways that you can do it. You don't have to adopt this method, but it is ways that you can do it. So the traditional serial process is the site enters the data, usually on paper. Then the site enters the data into the EDC. And then there's on-site monitoring and informed consent review. And then finally, central data review. So as we said, 6 to 12 weeks. And that was the median. So that means that, you know, there were ones that were out farther as well. Now with our method, we want the site to enter the data electronically into eSource, whether it's an EDC eSource or another electronic system, so that we can begin to review that data. And we also want the documents uploaded so that we can begin right away doing central data review, informed consent review, and that we can very rapidly retrain and follow up. And one of the studies that we did a survey of the sites, and one of the two favorite things they liked about using eSource, one was that they didn't have to transcribe data, but the second was that they got rapid feedback. And so again, if you can't change the way your data is collecting, but you can start to review quicker, then you're automatically improving the process from what you have right now. Now, this is a picture to try to help you understand the different ways that that you can, and I believe should, or that we look at data. So if you think about, let's go down to the very bottom. So if you are a monitor, right now you're doing SDV and you're doing it probably with your EDC, but you're looking at like the inclusion criteria, then you're looking at the vital signs for visit 1, and then the vital signs for visit 2, and then you're looking at whatever visit assessments that you have to do, labs or whatever. You're looking at all of this data, but you're looking at it by data set, by visit. So it's very disjointed, where and difficult. So I have our team, who has also done traditional monitoring in the past, told us they spent hours just trying to, you know, make sure that the dates are right with the ConMeds and the AEs and the med history. It's very difficult to do. And so this is the traditional method. So it's easy to miss things. And this isn't about being sloppy. This is about the method that we have to look at that. The recommended way is to look at all of the data in a more comprehensive way, what we call a subject source data review. Then so this would be the site monitor would do this source data review, and then our central monitor does this review. So what we want to do is, when we think about reviewing a subject's data, we look at one subject at a time, one subject, one visit, one form at a time. What we want to do is look at all of the subject's visits, all of their forms together synthesized. And then what we want to do is say, let's look at the site performance across subjects. So are we, the data managers like to say, are we seeing a lot of 0s and 5s on vital signs? Are we seeing too much variability or not enough variability? So what we're wanting to do here is look, are we seeing consistent errors that are being made across that site? So could it be dosing errors? Could it be informed consent errors? Could it be other kinds of things that you're seeing by specific users? We want to specifically think that we want to look at that data. So I tell people you can't find what you don't look for. So if you don't think about what do I want to understand that the site may be making consistent errors on, and then finally, how is that site performing compared to the other sites? So we know that when we have a bell-shaped curve and we have normal distribution, so this works, say, for if you're going to have a trial with, I say more than 10, certainly more than 20 sites, is you've got 95% of the subjects are within 2 standard deviations of the mean. And then you're wanting to look at those outliers on both sides. Why are they outliers? What are you seeing? Because this is an indication to you that their performance is different. So that is why it's important to look at these, because they're going to give you other indicators that you may miss. So let me just give you another example. We had a study where there were a lot of deviations at a site, and the monitor was spending a ton of time making sure that she got the documentation correct on the deviations, that it was appropriately reflected everything that you needed to do. But what she wasn't able to do was didn't do was to step back and say, hey, this site has a lot more deviations than others. And part of that may be she didn't know what the other number of deviations were across all the other sites. But that's what we're talking about, is we want to be able to look at these kinds of performance indicators across sites, because that's starting to tell us, oh, spend more time here. And that may be more time because of the site. It may be more time because of the monitor. It may be more time because they're sicker patients. There's lots of reasons for it, but you really want to understand that. So I think that this is an important slide to help you start to think about how you're going to look at quality oversight for your trial differently. And then I just wanted to give you a little more detail of how we do this. Again, from a timing standpoint, one is to start with immediate data management reviews so that we know if there's missing data, missing assessments, if there's problems with the key efficacy endpoints, if there's problems with dosing. So that DM review is going to result in deviations, it's going to result in queries, it's going to result in a number of these it's going to result in issues into our issue management report. And then at the same time, which I know is different, but the monitor is reviewing the data in a different way. So they're looking to make sure that the site monitor is making sure that that's a correct subject, that the inclusion exclusion have been met as much as you can do, perhaps remotely. It may include bringing some source in. They're going to look at whether we call this a triangular review or rectangular review, AE, ConMed, Med History, and Labs. They're going to look at a source data review using what we call our SPAR, which is our Subject Profile Analyzing Risk. They're going to look at GCP compliance, look at document review, including informed consent, IP management, and safety. So they're looking at data a little differently, but again, that will feed into deviations, enrollment rates, screen failure rates, making sure that all of these kinds of reviews feed into this information that is then reviewed by the central monitor. And the monitor reviews data again across subjects at a site and across sites. And then once we do that, we provide a total report on the health of the study on a monthly basis to the sponsor. And we also provide progress reports to each PI for each site, so that when the site monitor talks to the PI, the site monitor can provide actual performance data. And we have found that to be a much more powerful way to have a very productive discussion with the PIs. So thank you very much for participating in this webinar. As I said, you'll be getting a survey that has some questions in it, and also it'll include the link to the next webinar. You can also find that webinar on the manaRBM.com website. If you go under news, you can sign up for all the webinars there. And we'd love to hear from you. So if you have lessons learned that you want to share, please send me an email. If you have questions that you want or topics that we haven't covered in our list of topics, let us know, because we're here to help advance risk-based monitoring and advance quality in clinical trials. Thanks again, and I appreciate your attention and look forward to hearing from you further. Bye.